Way there goes. Screen down, sorry. We're gonna get started. Does anybody have any questions about anything before we get going with this? All right, beautiful. All right, so we're gonna do our chapter 5 Kahoot, then we'll do the chapter 5 activity, and then chapter 7 Kahoot and chapter 7 activity. You guys have any questions or any of that? You know, I'm gonna be calling on you just so that everybody knows what's going on. We've done it before, but I know it's been a while, so that's gonna be your routine. So we're gonna get started with chapter 5. For chapter 5 is discussing the eukaryotes. Remember what eukaryote means? True nucleus. These are organisms that have a true nucleus. Question number 1. Which of the following is found in a eukaryote, but not in bacteria? I mean, it kind of gives it away on the picture, but that's okay. All right, so this is the nucleus. So first person I'm going to be calling on is Kaylee Jackson. Is Kaylee here today? Okay, Kaylee, go ahead and tell me, what is the cytoplasm? One of the features of both of these cells. We know that that the nucleus was the only thing that the eukaryotes have that the bacteria didn't, right? That's that membrane-bound structure that contains the genetic material. But bacteria and eukaryotes both have ribosomes, cell membranes, and cytoplasm. So you want to tell me about any of these three, what they do for the cells?
Well, the nucleus holds the DNA,
Mm-hmm.
and then the cytoplasm is like the shape.
So the cytoplasm is just the goo on the inside. So basically, whenever we create that membrane around the cell, that everything in there is contained within goo, essentially. It's cytoplasm. It's mostly water. So say it's goo, but it's water with like electrolytes and proteins and sugars and things like that for the cell. So everything inside the cell, it's not air or a vacuum or anything like that. It's got actual stuff in there and the cytoplasm. That's fine. We're thinking about that process, so I'm gonna give you credit for that one. All righty, next question. Right. This organelle turns chemical energy into cell energy. Which organelle does that for the cell? All right, so most of us know this one. It's the powerhouse of the cell, which is the mitochondria. Jeremy, right? Jeremy. All right. Your question is going to be, what is that chemical energy? You remember what the molecule is that serves as the energy for the cell? Okay, that's fine. Do you know what the ribosomes do? Or I would say the cell membrane, right? Like that one's a pretty much a given. Okay, we'll come back to you, Jeremy. We'll come back to you with another question. All right, moving on. Oh, I will say this. Does anybody else know what the molecule is? Yes, ATP. So Dante is going to Deontay. Awesome, thank you. There we go. Oh, there we go. Next question, moving on. What is the function of the endoplasmic reticulum? Endoplasmic reticulum. So in the cell, we're going to be going over the processes of the cell and how the cell basically performs its functions and how that's controlled by our genetics in chapter 6. Understanding the basic parts of the cell and what their structures are can help us get into that mindset of how it works. So the correct answer here was that the endoplasmic reticulum is the site for protein production. All right, so one other popular one was controls what enters and exits the cell. That green one there. So that one is actually going to be our cell membrane. Okay, so cell membrane controls what enters and exits the cell. Stores water and waste. All right, does anybody remember what that? There is a part of the cell that does store water and or waste and other components in the cell. Does anybody remember what it's called? The vacuoles. Exactly. What's your name? Jordan. Exactly. Okay. What else do I want to say about this one? We know that the mitochondria obviously produces energy, so we're good on that, not choosing that one today. Righty, so I'm gonna go to kind of summarize these guys on the board. Endoplasmic reticulum is where we make protein. We make protein and other molecules for the cell. This is the manufacturing zone of the cell, whereas the Golgi apparatus, which is probably another question coming up, but that's okay, is the packaging and shipping.
Yes.
Yes, exactly. The UPS of the cell is our Golgi apparatus. And then we have we'll put things in vesicles in the Golgi, but then those vesicles might become storage containers, which we would call vacuoles. Vacuole storage. All right, lysosomes, which are vesicles also, but they're not vesicles for storage necessarily. We'll really store something until we need it, and that is our digestive juices for the cell. Those lysosomes are for digestion of the cell. for the whole body of the cell. All right, and there's other ones that we'll probably get to here in a little. We put mitochondria too. Mitochondria, energy, energy as ATP. We'll get to in chapter 10 in the next few minutes. All right. Question 4. What are the organelles that contain chlorophyll? All right, so these are the chloroplasts. So chlorophyll is a pigment that is excited by the sun. And when it's excited by sunlight, photons hit it and cause that to get excited and releases electrons as a result of that excitement. The electrons help power an electron transport chain, very similar to the ones that we learned about in aerobic respiration, cellular respiration. And let plants make ATP, basically without having to have molecules be broken down. We can get electrons just from exciting things with sunlight. It does not make very much ATP when they do that. When we're talking about cellular respiration, making like 34 ATP by breaking down a glucose molecule. And this, in photosynthesis, we're only making a couple of ATP, but it's enough. It's enough to get some ATP. We're in the sun all day, just building up ATP during our light processes. And then during our light independent processes, we call them the dark cycles, also known as the Kelvin cycle. They take that ATP that they made and they take carbon dioxide and they fix it into glucose. Now, I hate to break it to you guys. This is where a lot of people get disconnected. We're not teaching plant biology in this class, but some bacteria do this too. So it is relevant that we learn about this, okay? But in eukaryotes like plants, whenever they go through photosynthesis, that glucose that they made by taking carbon dioxide and fixing it into glucose, that's what they fix it into. What do they do with that glucose after that? They feed it into mitochondria for aerobic respiration. Plants need air too. Do they make oxygen while they're making that stuff? They make oxygen, sure, as a byproduct, but they're going to use oxygen too. They're going to make more oxygen than they use. And that's why we think of them as oxygen producers, but they need oxygen too. And so can't just keep them in a vacuum, right? First of all, they need the carbon dioxide access, but they also need oxygen in order to create ATP through aerobic respiration. Plants have mitochondria. They do the same exact thing as our mitochondria do. Okay. All eukaryotes have mitochondria. All right, that's the chloroplast. We talked about that Golgi apparatus just a second ago. We might have more questions about it. What do I want to say about this? We don't have questions about this. Chloroplasts. Okay, so chloroplasts are membrane-bound organelles that we find in some eukaryotes that undergo photosynthesis. So my question is for you guys. And again, anybody that hasn't answered a question can answer this question, and I'll give you credit for it. This is a difficult one, okay? But can bacteria undergo photosynthesis?
Yes.
Yes. So I kind of said it a little bit, right? Some bacteria do, right? So what were you going to say?
Isn't it like cyanobacteria?
Cyanobacteria, excellent. So yes, it is. Yes. Okay. Yes. So it is. Let's put this in perspective. They can't have chloroplasts. Chloroplasts are membrane-bound structures. Bacteria don't have any membrane-bound structures. Not a nucleus, not an ER, not a Golgi, not a vacuole, not a lysosome, not a mitochondria, and not a chloroplast. They don't have any of this. But they can still go through most of these processes that these things do. They can still make protein. They can still shuttle what they make throughout the cell. They can still digest things. They just don't store their enzymes in lysosomes. Okay? They just don't have compartments, basically. And so bacteria, when they undergo photosynthesis, write that on board. Bacteria, when they undergo photosynthesis, those pigments aren't found on a chloroplast. They're found on their cell membranes. That membranes is where they're found. Okay? That's where also their electron transport chains are found, in their membranes. They don't have mitochondria. Do they still make ATP? All right. Yes, they do. All right, beautiful. All right, so we're getting there. What was your name?
None.
Okay, thank you. I was like looking for it on here and I hadn't made it through the list yet. Okay. All righty, next question. Question 5. Doing great so far, guys. Waking up. Yeast are fungi. Is this true or false? Yeast. We know we get yeast infections. We don't get skin infections that are caused by yeast. We know you can grow bread with yeast. You know, you can make beer with yeast. Yes, anytime we're talking about yeast, all yeasts are fungi. We call them budding fungi or budding yeasts. They don't create the hyphae. They don't create that little branching. They might have pseudo hyphae, but they're still budding yeasts. And so we had that discussed in this chapter as well. Let's see. Question. Jeremy, I'm gonna come back to you this time. Can you give me one example of a disease that is caused by a fungi that is not caused by a yeast? Any general. Caused by fungus that is not a yeast. Like so excellent, beautiful. Yes, so that's an example of that, right? So we know we talked about yeast infections or skin infections, even ear infections can also commonly be caused by yeast. And those are fungi, but there's other fungi that can cause diseases as well that we will be learning about and talking about in detail. Athlete's foot is one of them. Athlete's foot actually falls into the category of ringworm. So ringworm is actually our cutaneous mycoses. Mycosis is any disease caused by a fungus and by molds especially. All right, moving on. Great, excellent. All righty, next question. True or false, fungi have chloroplasts and can perform photosynthesis? All right, so this one's false and most of us got this. Excellent. All right, so fungi don't have any chloroplasts. Is Kaya Hughes here today? I don't think she's here. She's back in the front usually. All right, she's not here yet anyways. Catherine Fields. Catherine, okay. So fungi don't perform photosynthesis, right? Can you tell me, so there's probably a question coming up, I'm probably ruining it. Fungi often break dead things down. Do you remember the term for that? So decomposition, but organisms that perform decomposition have a special term. Starts with an S. You remember what it is? I'm going to give you points either way, but just for calling out decomposition. There it is. Yes, saprobes. And that is going to come up pretty regularly. So we have fungi that might cause diseases that are often opportunistic pathogens, but our saprobes are the things that break down things that are already dead, right? So those are an important part of life cycles. We need to be aware of those guys too. Now, some fungi might be able to break down dead things, decompose things. Those saprobe organisms could also potentially have opportunistic infection abilities as well. So even though they're primarily saprobes, doesn't mean that they only are ever saprobes. They can also kind of switch between. All right, next question. Excellent. I think it's a question that's coming up, but that's okay. Do algae help to provide oxygen to the atmosphere? This is a little bit subjective, but not really if you read the chapter. All right, so this one is a lot, actually. And so some people put a negligible amount. All right, so Amna, this is your question. What are the primary producers of oxygen in our atmosphere today, generally speaking? So microbes, so going to be the eukaryotic microbes, but microbes in general are our primary producers of the atmosphere. When we actually calculate up, maybe when we talk about algae, all right, so these are eukaryotic organisms, right, and they are photosynthetic. Do you remember the family that they fall? I'd say the family, kingdom that they fall into. So when we have, basically we have 5 kingdoms, all right? So we have Monera, which is one that we are never going to talk about in this class. We're leaving the Monera out of things, okay? We're not talking about Monera. All right, so when we talk about the other 4 kingdoms that we do talk about, Animalia, Plantae, fungi. And I'm trying to think of the other one. What, sorry, what'd you say?
Sorry, I just, the,
Protista.
Yeah,
There it is.
that's
That's, yeah, it's protista. Yes, and so algae fall into protista. I was like, it's a P, it's a P. And I kept thinking protozoa, which is a problem that a lot of us probably will have on the test. We'll remember the protozoa. But which of those kingdoms do the algae fall into? Protista. They're not plants. Algae are not plants. I want to make that very clear. They're not plants. The plants have defined tissues. Algae do not, and that's the big difference with them. It also has a lot to do with their cellular structure and their genetics and things like that. But the algae are not plants, and algae are primary producers of oxygen, as well as things like plankton, microplankton, and things like that. Those are often going to fall into our protista category, or just small animalias as well. Okay. Another thing to remember is the cyanobacteria, like we talked about before. We have so many bacteria in the whole world, most of which don't even cause disease in organisms like us, just generally speaking. The cyanobacteria. So actually, a large majority of the oxygen made on our planet is not made by plants, not by the Amazon rainforest. That is nothing compared to what the algae and the cyanobacteria make. So microbes are the primary producers of oxygen on our planet, not even plants, not the Amazon rainforest. Yes.
Like, why are people freaking out?
Because you don't know better. I mean, that really is why. Now, does it mean that if we lose a lot of our plants, that we won't be losing a lot of our oxygen source? Yeah. I mean, plants still contribute a lot. It's just not the majority. And whenever we're talking about global warming affects our algae and our cyanobacteria and things like that in our marine environments. We have microplastics and things like that. And plastic is building up. There's an island of plastic. We're going to talk about this in chapter 9 in unit 2, but there's an island of plastic in the Pacific Ocean, kind of near India. It's, I think, in the Indian Ocean, but it is the size of Texas. It's an island of plastic the size of Texas. And this is not, it's the only, island of plastic out there in the oceans. So you can imagine, I mean, consider how long it takes for you to drive across Texas. That's how big this island of plastic trash is hanging out in the ocean. And there's multiple islands out there, but that's the biggest one, okay? And how that can create a massive change in the organisms that live around where that island is. You're blocking out sunlight with that plastic or changing how the sunlight behaves in those areas. You're creating weird surfaces for microbes to attach to that wouldn't be there otherwise and stuff like that. So we're changing our earth with these things, right? And over time, that creates a massive change in the types of organisms that we could see. So we might have a decrease in our oxygen producing bacteria as a result of climate change as well. So not just our trees that we're cutting down. So it's a big deal, the trees. And the trees also, we also got to think about with the Amazon. So why else will we make a big deal about these things? How many drugs and things like that were adapted from plants and stuff like that we found in the Amazon and places like that where we're discovering new things. So diminishing microbial or not just microbial, but the ecosystem and the diversity of our ecosystems, macro and micro, is the result there. All right. Question 8. These single-celled organisms have trophozoite and cyst life stages. Trophozoite and cyst. These terms are specific to exactly the protozoa. All right. So our next person that I'm going to call on, I'm assuming that Saber Thompson Hale has not shown up for class still. Yes, I'm assuming that's somebody I have to withdraw from the class. All right. London McNair. London. Okay. All right. My question for you is trophozoite and cyst. You can pick one or the other. Equate it to similar structures that we talked about previously with the bacteria. So one or the other. I don't care which one though.
Can you restate that?
Yes. So when we look at trophozoite and cyst, these terms relate to a process that we talk about in bacteria. So you can talk about the process or the name of the structures. What does trophozoite relate to? Or what does cyst relate to in bacteria? Not quite. Close. We're in the realm of it. We want to be more specific than that. So it's specific trait. So protozoa organisms can cycle between trophozoite and cyst forms. And this is similar to something that we talked about in the bacteria. Yes.
Active.
Close. We're getting there. We're getting there. I know Deontay knows. I know you guys know. We already called on you. John. Endospore. Okay, so we'll get John. So we'll get you some points there, so I won't call on you later. We'll come back to you, London. Okay. No worries. All right. So endospore. So that endospore cycle. Remember the endospores. I talk about them a lot, and we're not going to stop talking about them. So the endospores, those hardy, completely dormant, completely inert structures. Some bacteria, Bacillus and Clostridium bacteria, right? Okay, we're going to recall it, right? That they can go completely dormant and make these endospore structures, right? Protozoa can do something very similar. And when they do that, it's not endospores because these are eukaryotes. I mean, they're doing something completely different. But it's a similar process, and we're not going to break them down and really like nitty gritty compare and contrast. But it's similar enough. We can say cysts are essentially the same thing as an endospore. They're completely dormant, they're inert, but they're incredibly hardy. All right. Anybody ever hear about if you go swimming in swimming pools, the likelihood of you getting sick from the water in the swimming pools? Yes. And people are going to the bathroom in swimming pools. So we know you're peeing in it, but I'm talking about poopy stuff. Okay. So people get a poopy disease in the water. We're hoping that the chlorine will take care of it. There's a type of poopy disease caused by Cryptosporidium. And Cryptosporidium is a type of organism. It's a protozoa, and it makes cysts. Obviously, protozoa, only protozoa make cysts and turn into trophozoites. And those cysts are incredibly resistant to chlorine. The trophozoites aren't. The trophozoites get killed. The cysts, the dormant, the hardy versions of them will be able to survive the chlorine.
Wait,
Yes.
is this the same cyst as like this?
No.
Okay.
Yeah,
Oh, well,
so like
that's just
skin
better than
cyst,
my
yeah, no, it's completely different from like a skin cyst or something, like that. So when you get like a cyst, like we think of a human having a cyst, we might think of like cystic acne, or we might even think about in diseases, like certain diseases, like with tapeworms, for example, which tapeworms are not protozoa, right? They are animals, right? But they can encyst, ENCYST, and they create little big old balls inside of like your muscle tissue and stuff like that, or in your brain or whatever. They do that in your brain. We call it neurocystosarcosis.
So we can get to the branchials.
So again, sporangiospores and the cnidiospores, those will be the fungi.
Oh,
Okay, yes. So categorizing those guys based on their reproductive stuff, which I don't think is in these question reviews, but do know the difference between a sporangiospore and a cnidiospore. That is a question that is going to appear on both the lecture exam and the lab exam. Okay, just to review that. I don't know if it's going to be in these questions or not, but I might as well review it now while I'm thinking about it since Deontay brought it up, right? Sporangiospores or sporangiophores, okay, the actual structures that are making that we would call, they look like flowers, sort of, okay? Sporangiospores are the spores that they release. Sporangiophores are the organisms that make these structures. So it doesn't really matter, and I'm not ever going to make you tell me the difference between those two terms. But sporangio and cnidio, we should at least be familiar with, okay? So cnidio, these are for fungi. And how they reproduce asexual. This is the reason why when you have your bread and it's turning green, it can cause the other bread in the next package over to turn green. Okay, because they release these asexual spores and they can flutter through the air and whatever. So sporangiospores, when we create those kinds of spores, these are spores. These are not endospores. These are reproductive spores. Like when you think of fungal spores, that type of spore, okay? When you have a cover, it's a sporangiophore, sporangiospore. Like I said, I'm not going to differentiate between spore and spore. I'm not. You can remember sporangio is the covered ones, and cnidio are the open ones. They look sort of like this. They can look like dandelions too. They don't have a cover around them. All right. The one that we commonly will see, it looks sort of like this. It looks like a little paintbrush sort of coming off of it. Okay. One that is shaped like that, that's penicillium. So penicillium is one. Aspergillus, another really common mold in our environment, also does not have a cover around it. It looks like this, but without, it's like a dandelion, but no cover on it. And I know dandelions don't have covers, it's just like a dandelion. But sporangio guys have a big cover around them. And so they'll like grow the spore inside and it bursts open and releases all the spores. Okay. These guys just are growing and they're open all the time and just release them as they go. Okay. So there are two different types of asexual reproduction for fungi. So that's what these are. Okay. All right. We were back at tropozoite and this really quick. For protozoa. And then for bacteria. We know that bacteria, if you are hardy, resistant, and inert, the dormant one, all right? For bacteria, that's the endospore. For protozoa, that's the cyst. If you are active and multiplying, for the bacteria, this is the vegetative cell. And for the protozoa, this is the tropozoite. Probably not going to remember my example for how I remember this. But when I say trophozoite, we're going to be talking about a lot about autotrophs and heterotrophs and chemotrophs and phototrophs and whatever. Troph means eat or food. I also think of trough when I see it. It's like a trough that pigs eat out of. Okay. If you're eating, you're active. If you're inert or dormant, you ain't eating. Okay, so that's how I remember the tropozoite is the active one. Maybe that helps, maybe it doesn't, but whatever. For bacteria, only Bacillus and Clostridium species can switch between endospore and vegetative. Those are the only ones that make endospores, all right? For protozoa, it's a ton. If there's a lot, we're not going to name them. Most of them, right? Not all, but many of them can switch between tropozoite and cyst. Yeah.
So there are, there are like probiotics that are spore-based probiotics.
Mm-hmm.
Does that mean they're only bacillus-based probiotics then?
Yeah. So they'll either be called bacillus something or lactobacillus. It has bacillus in there, so that counts, right? So yeah, so most of those you'll notice if you read the back of your bottles, many of them are lactobacillus, and many of those species are actually good enough. When you take a probiotic pill, we know that they have a lot of bacteria, and you probably read the back of them, seen all those bacteria species that are on the back of them, yes? They are not permanently incorporated into your gut. It's not like you take it and then now your gut has that forever because your stadium seats were already full. There's nowhere for those new good guys to sit down either. Bad guys can't sit down, but neither can new good guys. And so that's why you have to keep taking probiotics. If you take antibiotics though, and then take some with probiotics, then potentially you could be filling up the stadium seats with the good guys. But that's why it's so important. We'll say when you finish antibiotics, eat yogurt and stuff like that, because those cultures can help to repopulate your gut microbiome. We actually firmly believe that your appendix serves as a source for your gut biome, so that if you were to deplete your seats, open up your stadium seats by taking antibiotics, that your appendix is there to hold on to whatever your biome was previously to help reseed your microbiome when you take, not necessarily antibiotics, that's not what we've evolved for, but anything, stress, something like that to help repopulate your gut microbiome.
What happens if your appendix bursts?
Yes, so that's why the deal is, that's what the problem is with appendix getting infected and bursting, is that those microbiomes that they're holding on to, we think are overgrowing for whatever reason. That leads to infection, which we know now, I'm sure you guys are familiar with, when you have an infection like your appendix has become appendicitis, right? It becomes inflamed. It's usually the overgrowth of bacteria in that appendix that is causing that inflammation. Then your neutrophils come in and try to eat up all that stuff and control it, but it's not able to because there's just so many bacteria. And then it can rupture and it releases pus into your abdomen. And that's going to be the pus is primarily neutrophils, but it's also largely, other than the neutrophils, the bacteria, clearly. And that doesn't belong in your gut area. It belongs inside of your colon, but not in your actual gut, outside of the gut, like outside of your intestines, right? Bacteria belong inside of your intestines. But if I were to take a swab, cut you open and test around your intestines, not in them, but in the actual gut cavity, it should be sterile. So when we introduce that stuff into that area, we're talking about an open stadium now that becomes very, very quick and easy for microbes to cause infection and disease after that, which is why it's such a serious thing when that appendix ruptures. Luckily, if we can catch it early enough, obviously we can treat it. That's an issue, but yeah. All right, moving on. Any other questions? Okay. Question 9, what are helminths? From these choices, which are the helminths? 2, 3, 4, 5, 6, 7, 8. All righty, so these are worms, right? So specifically parasitic worms. So we call them parasites. Why? Okay, let me see. London, since we didn't quite get you on that last one, right? So we'll come back to this one, okay? This one shouldn't be too difficult. Why don't bed bugs fall into the helminth category? So if the helminths are parasitic worms, how would you classify bed bugs? They're not one we talk about in the chapter. I'm just asking about general knowledge about a bed bug. Don't think too hard on this one. Yes.
They're not parasitic.
They're not a parasitic worm. Don't think too hard on this, okay? So bed bugs, what would you classify them as outside of this classroom? What would you say they were? Bugs, right? And so how do we call bugs? Insects. Okay, there we are. Okay, right. So insects. So can insects be microscopic? Right. But these are not helminths, right? And so bed bugs obviously are an insect that will bite you and feed on the blood of whoever they're biting. It's the same thing with scabies and lice. These are insects. And so if you know scabies, then you know this scabies is very similar to like mange in dogs. Oftentimes mange is very similar.
It's awful.
Yeah.
We haven't had like scabies that much in the ER, but we've had bed bugs before. And literally, it's like we're putting on spacesuits to go into the.
I'll tell you what, it's like I saw this meme on Instagram, but it's so true because I worked in a hospital. I worked in the lab, which was removed from the patients. But at the same time, when there was bed bugs in the hospital, it was like all hands on deck.
Oh,
Like,
yeah.
You look in the corners, you
Oh,
look
yeah.
everywhere, you like inspect everything. If somebody's coming from a patient area, then we will clean the areas that they could touch and stuff like this. Okay, bed bugs are pretty small, but you can see them with the naked eye. But that's exactly, they almost never go away. It's sort of like once you have them, it's almost impossible to get rid of them, especially in a hospital, you guys. And so you will work in hospitals and there will be bed bug outbreaks. You may or may not know about them, but they are there.
We
I'm-
spray from every night.
Yeah, I'm telling you, it is not uncommon.
Wait, do they like cover on people's bodies?
Yeah, usually,
Yeah.
usually the patients bring them
Patients Bring
in.
them in and
Mhm.
it just spread like wildfire.
Yeah.
It don't ever end.
No, I remember I was at Integra Southwest and I don't know, I think it was a patient that came in for a blood draw or something. And I remember like they came in and when they left, there was a bed bug crawling on our like desk or whatever, our countertop. And I mean, it was full blown, like we took everything apart, like the whole desk, we took it apart and cleaned up it all. And like, we had called up like maintenance and stuff like that for them to come and see, like we have a bed bug up in the lab area. And whatever. And so it's just uncommon to see it in a lab because we didn't have as many patients up there. But they would make it up there. And I remember that the whole problem in that building at that time was radiology had bed bugs. So
Everybody, yeah.
My favorite is when,
everybody did, yeah. And it was like literally you go home and you take your scrubs off because we wore scrubs in the lab too.
You take them off in the
Take them off in the garage and you
wash
put them in, you either wash them or you can just put them in the dryer too. If you want to just dry them. I would put mine straight into the dryer and my shoes too for 30 minutes on high.
Yeah,
Everything went in there and then I'd go take a shower immediately before I,
my favorite.
it's the real life of little doing.
The doctor spread.
Mhm. Yeah.
My favorite thing is when like a patient is like, you've been in and out of their room all day and they're like, oh, by the way, at my house, I have bed bugs. Great.
Yeah.
Thank you.
Yeah.
man.
Yeah.
Thank you for telling me right now.
So one thing, we're pretty familiar with bed bugs and the risk there. They're really, really hard to get rid of. And if you are ever suspecting that, for example, they can be kind of rampant in low I'm assuming rent, that's not what it really
Like
means.
low
but-
income.
Low income areas, especially like not just in their own housing, but in like motels and things like that that are associated with that kind of level of existence. So if you ever stay at a motel like that, check. And I don't mean that look around vaguely. I mean like look at in the actual seams of the mattresses and things like that. That's where they'll hide in the nighttime, and then like you'll sleep on the mattress, and then your carbon dioxide draws them out like mosquitoes draw. And then they'll come and feed on you, and you'll only have like a couple of like itchy spots in a little cluster, but that's the sign that there's bed bugs there. And then they can travel back home with you. So it's really a big issue with them traveling back home with you. Uh-huh. So again, we don't talk much about the insect diseases, but just for awareness's sake, okay, guys? Scabies is pretty hard to contract from another person unless you're in close contact with them regularly. So we have had issues. I said when I worked at Oklahoma Blood Institute, my husband still works for them. He worked for them for 15 years. But we would get people coming to donate blood, and they'd have a rash on their arm. We go, "What's that rash?" And they're like, "Oh, I have scabies." And we're like,
Who just cut?
so, remember those questions that you're supposed to answer before? Like, that's part of what you should do. When you're feeling unhealthy and well today, one of the questions to ask you, are you feeling healthy and well today? If you have a rash on your body, you answer no to that question. That's a no. Okay, so just to be clear. All right, that's a no. So we had to go back and change the question. That happens all the time, you guys. All the time. Multiple times at least a year. Maybe not every day, but regularly enough. Okay. And people have scabies and things like this, and they're just like, whatever, I have scabies. You can go to a doctor and you can be treated for that. And there's ways you can treat scabies that is just very manageable. And again, though, just from touching somebody that has scabies doesn't mean that you're going to get it. It usually requires a little bit more contact and close environment than that. Most of us are probably familiar with lice. Most people have probably had lice at least at some point in your life. Most females probably have any. If you went to daycare like I did, you had lice. Okay, that was just the part of the ritual. And it is also manageable. There are plenty of medicated shampoos and things like that that can help eliminate lice. However, if it goes unchecked, that's whenever it becomes an issue. We start having overgrowth. And it's like if you know, you know, or if you have kids, then you know, right? If one person has lice, the whole family has it. I don't care if only one of your kids has an itchy scalp. You all have it. For lice.
Because other kids like stereotypes.
Yeah.
So if they find out they can contact the parents have the option to come get them. But they don't. Yeah, they
Huh.
don't have to sit them home.
Wow, whatever.
Yeah.
I mean, I went to daycare enough years as a kid. And if you have kids, then you know, okay? It is super common. It's super common. And it is very manageable, but it's also one thing. Now, having dirty hair actually makes you less likely to have lice.
Yeah,
Having
my son
clean
has
hair.
a head full of hair, like he normally wears an afro, and I've had so many calls of another kid having lice, but I would go through his hair and he would
Yes.
never have lice.
And so, as I'm sure most people know, is that, people in the Black community often oil their scalps and have like lots of conditioners and things like that, and don't wash their hair every day. Those natural oils make it harder for the lice to live, and they can't attach their eggs to the hair shaft and stuff like that. So the cleaner your scalp is, the more likely you are to have lice, actually. Put that in perspective. Learning things that are, I'm not going to test you on that, but I feel like it's useful to know. All right, what are platyhelminths? Platyhelminth. One, two, three, 6, 8. Righty, so these are our flatworms. Jimena, are you here today? Okay, so that's you. Okay. All right, so nematodes, what are those ones? You're all right to look it up in your notes. So the helminths have a lot of fancy scientific names. They can't just have made it easy for us. And so I'll kind of write them. I'll write them on the screen. Exactly. Good. So I was buying you a little bit of time there, but yes, exactly. They are the roundworms. So with the parasitic worms, let's put worms, but these are our helminths. These are parasitic worms. Okay. Not all worms fall into this category. Earthworms, for example, they don't fall into helminths. Okay, they're called annelids, but they fall into animalia too, but they're just not helminths, all right? So our helminths, our parasitic worms, fall into two groups, the roundworms and the flatworms. The roundworms are called nematodes. They don't have any subcategories, but the flatworms do. Flatworms are called platyhelminthes. Flatty makes me think of flat. That's how I remember the difference, okay? So our general term for our flatworms are the platyhelminthes. These are the flatworms. There's two categories of our flatworms. These are the flukes and the tapeworms. All right, the tapeworms are called cestodes. Cestoda, cestodes, however you want to talk about it, but these are tapeworms. And then our flukes are called trematodes or trematodes. Trematoda is what the group is called. These are flukes. You may not know much about the flukes, and probably something that you've probably never heard of or heard much about. But if you were like me and you liked to watch the show Monsters Inside Me or whatever it was called, freaking loved that show, okay? But it talked quite a lot about things like liver flukes and blood flukes and things like that. They are small, whereas we think of a tapeworm as being a long, big old tape of a worm. Some of the tapeworms aren't quite as long as those ones, right? We know those can be like meters and meters long. But the flukes are like tiny little short flat guys. They remind me of, like, you guys know the difference between rays and skates. Probably heard of a skate, but they're like a ray, but they have more of a shark shape, but they're like a flat shark. That's kind of what the flukes look like, right? But they're very, very tiny. And they'll live in your bloodstream, in your spleen, in your liver, and in your tissues in your body, just like the worms can, okay? But we'll talk a little bit about those diseases when we get into later chapters, all right? So good job with that. So yeah, technically nematodes and roundworms, same. All right, let me remove this. Next, here's our little podium for this, and then we're going to get into the chapter, whatever this is, chapter 5, case study thing. Good job. All right, we'll leave that kind of up in the background. Is it gonna start with the? You usually ask me if I want to start another one. It's just taking its time. There it goes. Okay. So we'll leave this up in the background, but chapter 7 is what we'll be doing next, which is the viruses, right? So we'll come to that in a little bit. I'm going to start it, but you don't have to have joined yet. And we'll just come rejoin after we do the activity, but I'm going to have it in the background. So it's there. All right, chapter 5. Again, chapter 5 was our eukaryote chapter. And so this is talking a little bit about disease associated with the eukaryotes. And as it happens to be, this was talking about a disease caused by a protozoa, Plasmodium falciparum, that causes the disease malaria. So there are a lot of different Plasmodium species that can cause malaria. Plasmodium falciparum is like easily the worst one that you can have, the worst version of malaria you can have. But there are other ones like Plasmodium vivax and Plasmodium malariae and all sorts of other ones that are associated with malaria, generally speaking. Okay. Yes, you know about malaria. Cannot be stated enough how much of an issue this is worldwide. It might not be an issue here in the United States, but again, our perception of what a problem is in the world is very gated compared to the rest of the world. And many of these diseases that we're going to talk about in class, you'll see what I mean. But anyway, so this is malaria, and malaria is rampant in our tropics. Okay, so it's not just, we know of malaria being in sub-Saharan Africa, where it is a huge issue in sub-Saharan Africa, but I want to be very clear that malaria is an issue in Asia as well, and as well as in South America and Central America. So we do have malaria risk even in Mexico. There's malaria. And there's certain states in Mexico where if you've traveled to it, you're not allowed to donate blood because the malaria risk. So stuff like that. So we want to be very clear about this, that malaria is not only found in sub-Saharan Africa. It's found in a lot of different parts of the world, all right? So this was our, basically it's an overview of cases of malaria associated with this. Physicians in the US do not frequently encounter patients with malaria. Most of the time, the patients that US physicians see with malaria are people that traveled outside of the country and did not take what we call prophylactic medications. And prophylactic medications are medications that you would take to prevent malaria. And they actually happen to be the medicines that we usually treat malaria with. Just like with antibiotics, right? Sometimes whenever you're going in for surgery, we'll put you on antibiotics before you go into surgery just to prevent infection. It's the same idea here, all right? So this is talking about a previously healthy 55-year-old woman that had no health history when they presented to the emergency department in Florida due to a cyclic fever for 7 days. She had a fever and it would go away, and then she'd get a fever and it would go away. It seemed to be cycling. Patient returned from a 10-day trip to Ghana 18 days prior to admission. On arrival to the emergency department, the patient appeared lethargic and within hours was in respiratory distress. She was intubated and ventilated for respiratory failure. We looked at a smear of her blood and saw that there was Plasmodium falciparum in her blood. Now, Plasmodium falciparum, again, a protozoa, which is a eukaryote. It lives inside of your red blood cells. It'll multiply inside of your red blood cells and then burst out. Whenever it bursts out, it causes the fever. And so you'll have a fever whenever they burst out of your red blood cells, and then you'll recover from the fever. But new ones are growing in different red blood cells. And whenever those ones burst out, you get a fever. It causes massive inflammation in the body. It causes liver damage and spleen damage if you're rupturing that many red blood cells. They also live in what we call merozoites that are developing in your liver. So they can actually hide out in your liver for long periods of time, and can actually, even if you've had malaria and you've been treated, we still consider you a risk because we can't always treat it when it hides out in the liver that way. So it can be quite a difficult thing to get rid of.
You can't ever get blood again.
If you've had a positive for malaria, like if you've traveled to an area with malaria, then there's like a wait period. We want to be sure that you don't have malaria. Then you can come and donate. But if you,
When I was younger, but I haven't, like, I've been able to get blood since I've been in the US.
yeah, so there will be like time periods, and they can actually like look, like if you've been treated and you've been tested multiple times, and they can actually look at your personal history and like determine whether or not they think you're actually a risk still. But most of the time nowadays, they pretty much say, well, have you ever had malaria? And there's a series of questions that they'll go into if you have ever had malaria.
A lot.
Yeah, yes, there's a lot of questions for it about that. So if you have, and then depending on when you had it and how you were treated and stuff like that, they might determine if you can still donate or not. But many times it'll be like, oh, you've had malaria, well, you can't donate.
My daughter is 17, and last time she had it was when she was 12, and she still can't donate
Yeah.
because she had a situation at school when she tried to. and they-
Yeah, it'll be one of those things. I think it's a certain number of years you have to be negative for after, but I don't know much about that stuff. And they changed the rules with that too. But anyways, let's get to our questions about this case study. Which organelle in the pathogen that causes malaria generates the energy that powers all the cell's functions? Mitochondria. So it's easy enough. The powerhouse of the cell is the same for our malaria. It's all eukaryotes. Same. So mitochondria. The pathogen that causes malaria is a eukaryote. Okay, we know that because it has mitochondria. Why might this create unique problems when treating infections caused by this pathogen that might not be associated with treating bacterial infection? So it's okay if we don't know this right off the bat, right? But this is a critical thinking level of question. We can compare. So Deontay, I know you have a thought. Go ahead and share it.
Is it because that, with it being eukaryote, they're all membrane bound?
Yeah, so we can look at it that way. Think of it this way. Many of the bacteria infections that you're having, we can target peptidoglycan because your body doesn't have, your cells, your eukaryotic cells don't have peptidoglycan, right? Target that. That's a molecule that we know that, we don't have in common with them. Stuff like that. That, like we said previously, the folic acid synthesis pathway. I know we haven't talked about it much, but we're going to definitely get into it in Unit 3. Stuff like that. Those are easy targets because our cells don't do that as eukaryotic cells. But Plasmodium falciparum and other protozoa and other eukaryotic infections create a different kind of challenge, because now we have to find, we can't just target general aspects of these cells because we share the general aspects. We have all of these things in common. all eukaryotes will have these things in common. So I have to find other things to target with them. And if you target DNA synthesis or protein synthesis, then you're targeting your own cells in addition to these guys, which is the hugest, biggest problem with treating many of these eukaryotic infections, okay? Especially long-term. So luckily, we have found a few things that are unique between some of these bad guys and us, that things like fungal infections aren't too hard for us to handle those medications, right? Take ketoconazole or miconazole, you can buy those over the counter. You can buy ivermectin over the counter. Be very clear about ivermectin. Please do not take it over the counter unless your doctor tells you you should. I know there's a big thing going around of like pharmacy companies telling us that we have parasitic infections and that they've been hiding it from us and they want to keep us secret. That is not true. It is not true. Like it actually isn't true. Right? So don't buy into that TikTok stuff, right? Because ivermectin is actually pretty dangerous for you to take it. In fact, anybody in here work in a vet clinic? Okay. Have you had dogs come in with heartworms that have been on ivermectin? Okay. So sometimes we'll see that in vet clinics. We'll get, I want to be very clear about this. Heartworms, very common. Make sure that your dogs and your pets are on treatments that prevent heartworm. Okay. My dogs, spaghetti and meatball. Okay. So spaghetti and meatball, we adopted them contingent on knowing that they had heartworms. We adopted them and that we were willing to deal with the care for them while they recovered from it. And that's the place we adopted them from, which was Pets and People, paid for the treatment for their heartworms, but we had to like manage them while they were being treated because it is very dangerous to undergo treatment for heartworms. The treatment for heartworms is just ivermectin. Don't just take ivermectin. Yes.
What about topical?
So topical ivermectin will be for things like scabies. Okay, that's usually what that will be prescribed for. And as long as it's just on your skin and it's used for scabies or something like that, right? Or for like dogs, we'll see topical ivermectin for like mange and things like that. Okay. As long as it's just being used during those treatment cycles, it shouldn't be as much of an issue as whenever we take it internally. Okay. But we'll take ivermectin is usually going to be for the worms or the micro insect infections typically. Don't take it just because. There's also been, I'm sure you guys have heard about this on TikTok and whatever. If you're following health stuff like I am, then you've heard these things. But it was investigated whether or not ivermectin could be used for treatment of COVID. It actually was found not to be effective for treating COVID because it was more dangerous for people to take the ivermectin. Now, did it actually help with the viral infection? There was no clear indication that it did. Some people read online that, oh, this was a miracle cure that they were keeping from people, that you just take ivermectin and you'll get over COVID. Don't fall for stuff like that. Now, if you don't know how to tell the difference between what you're reading on social media versus what's actual facts, then I'll help you look for things like this. This is an actual medical journal that is peer reviewed by other physicians that, you know, other people have read it and said, yes, you can go ahead and post this because it isn't a load of BS. And so if you have a hard time knowing what's real on TikTok, then I'll help you find journals like this. It will help back up what is real so that you can find the difference. So we can stay informed with that. All right. Okay, just saying. All right. Next question. Is the pathogen that causes malaria multicellular or single-celled? Does anybody know this one? It is single-celled. What's your name? Okay. I won't call on you later though. It is single-celled, exactly. And this, we know this because all protozoa are single-celled. All of them are. None of them are multicellular, just single-celled. That's the defense of it, all right? It says defend your answer. If I had something like this on the exam, I might not ask about malaria. I would put some context in with it. I would tell you malaria was a protozoa. Is it single-celled or multicellular? Defend your answer. And you would say it's single-cellular because all protozoa are single-cellular. Okay. All right, pathogen that causes malaria, Plasmodium falciparum. Please excuse the yes, because when I wrote it, it was italicized, but then when I turned it into a PDF, it changed the formatting on it. So I apologize for some of that stuff. But it should be italicized. Plasmodium falciparum is a protozoa known as an apicomplexan. Now the chapter does talk about the differences in our protozoa motilities. There are 4 types of protozoa based on their motility. Yes, okay. So these are going to be, and I'll write this on the board so we can track it. Our 4 types of motility with protozoa. So motility. Yes, exactly. We have first one, flagella, easy enough. We learned about the flagella with bacteria. Now, they're more complex flagella. In eukaryotes, the flagella are bound by the membrane all the way through, and that eukaryotes can move them like whips, not just by rotating one way or another. It's much more complex. Another one is the cilia. These are tiny little hair-like appendages that move like oars. They sort of beat the usually water around them. These are usually for organisms that live in freshwater or marine environments. But in case you didn't know, for eukaryotes, generally speaking, you know about cilia because we have cilia in our own gut that are helping with absorbing nutrients. And so these cilia do the same thing for protozoa, which we're not protozoa, but we are eukaryotes. They do the same thing for helping with feeding and stuff like that, but can also be used for motility. Here's an example of some organisms that are associated with disease from these guys. One of the flagellated guys would be Trichomonas. Trichomonas. We've talked about a little bit about Trichomonas. Most common non-viral STI is Trichomonas. So it's a protozoa. Then we have cilia trick, they call it. Cilia, this would be like Balantidium coli, which is not a very common one, but it is one that can cause poopy illness.
And it's called celiatric.
Flagella, if it has one of the examples of a flagellated protozoa is Trichomonas. We call it trick as the disease, right? It is an STI. All right, I'm trying to give an example of each of these. There's plenty. Like we think of, you guys probably heard of Giardia, maybe. Okay, Giardia is flagellated. It's flagella. 3, Pseudopodia. If you have pseudopodia, pseudo is fake. Podia is like podiatrist is feet. So these are our amoebas. So let me put this. This is all of the amoebas fall into this. An example of an amoeba that causes a disease might be Entamoeba histolytica, in its name at least, okay, Entamoeba. Entamoeba histolytica or Entamoeba coli. But Entamoeba histolytica is associated with dysentery, which is bloody diarrhea, bloody mucousy diarrhea, okay? Another amoeba, obviously we think of the brain-eating amoebas, but you might not know their names. That would be acanthamoeba. Acanthamoeba is associated with the brain-eating amoeba disease or with keratitis, which is an infection of your cornea. Pretty common from contact users that don't use sterile saline. If you're using tap water, gotta stop that. Gotta stop that. Okay, you can go blind from that, and that's from acanthamoeba. You can also get the brain-eating amoeba from it. Another brain-eating amoeba is Naegleria fowleri. Both acanthamoeba and Naegleria fowleri, when they cause the brain-eating amoeba disease, we're talking 97% mortality. The 3% of people that survive of it, they're going to have serious brain deficits. Nobody just like recovers from it. And it happens quick too. Usually if you were to come into a hospital and we diagnose you with the brain-eating amoeba, it's going to be palliative care until you die. It's very serious, but extremely rare. Extremely rare. I think there was like 2020, there was like 13 cases or something like that in the United States. Pretty uncommon, all right? The 4th category is going to be the non-motile group. These guys can kind of move, but they are not actively moving. They might be using the body's systems to move around. And this group is called the apicomplexans. This includes things like malaria, right? Or trypanosoma, stuff like that. Okay, so this one's non-motile. So that is our answer for question 4. Tells us it's non-motile because all of the apicomplexans are non-motile. As it turns out, all of the apicomplexans are all parasitic. A parasite is something that harms the host when it gets benefit from the host. So one thing is being harmed and the other thing is benefiting from harming the thing. like we often think of ticks and leeches as being parasites, right? They're feeding off of you, they're causing you harm, and they're getting nutrients and stuff from that. So all apicomplexans are parasitic. Now, all of these guys that I named for you, the brain-eating amoeba, Phalantidium coli, Trichomonas, those are also parasites. But I'll tell you, there are plenty of other protozoas that fall into those other categories that just exist in the wild. They don't hurt you in any way, okay? So, all right, fifth question. Malaria is caused by Plasmodium species. SPP will always be species, okay? Plasmodium species are transmitted by Anopheles mosquitoes. The United States already has a significant population of Anopheles mosquitoes that could potentially harbor malaria-causing pathogens. Why would rising temperatures in the US be a concern? It's easy enough. As the tropics are moving north because of rising temperatures, so are the mosquitoes, and so are the mosquitoes that carry the diseases. In fact, we have had a couple of cases of malaria in the United States that we think might actually be harbored by mosquitoes here in the United States. We just haven't been able to prove that yet. But if there's mosquitoes in Mexico that have malaria, of course we could have mosquitoes come up into the United States. We have plenty of tropical diseases that we have not seen in the United States that are now becoming endemic. That means it's commonly found here in the United States. Back in our day, I say our day, but when we were younger, all of us, we remember the big scare that was associated with Zika and West Nile virus. Okay, we do know that West Nile virus hasn't gone away, right? It is still rampant. Most people, probably 90% of people that get West Nile virus will be either asymptomatic or subclinical. You wouldn't go to the doctor for it, but you might have a rash or something like that and be like, what's this rash from? And then continue about your day. West Nile virus is commonly associated with encephalitis, but it can be a low level enough encephalitis that you might just have a mild fever and a rash and then recover from it. And then you have immunity for the rest of your life after you recover from it. But some people develop serious encephalitis and can have serious illness. That might not sound like much or be very concerning, and that's why it's not, but we still do track it. And it is still a reportable disease here in the United States because some people do have severe effects from it. But some other ones would be dengue. Dengue, which was more commonly associated with South America and Central America, has now moved into the United States. It's now endemic here in Texas and Florida. So expect to see more people that have dengue fever. It is a hemorrhagic fever. It can present as either a hemorrhagic fever or very, very similar to malaria. We have a cycling fever that they also call it the breakbone disease. It causes severe like shivering and shaking associated with that fever that it presents with that can actually have been known to break bones in small children. So it's the breakbone disease, breakbone fever.
Are you saying dengue?
Dengue, D-E-N-G-U-E. It is a viral disease. It's not protozoa, but these are the tropical diseases that are now moving into the United States as a result of global warming. Okay? I don't care why you think global warming is happening, but if you're going to say that it's not happening, I'm going to look at you in the face and be like, come on now. Global warming is happening. I'm not going to argue with you why, but it is happening. Like temperatures in the world are rising. That's documented. That's true. That's proven. It is happening. Why it's happening, that's a different argument. Okay. But whether we're causing it or not, I'm not having that discussion in this class. Okay. You can have a discussion about that with your other professors, but it is happening. And so other diseases is one called chikungunya, another viral disease. Transmitted, all of these are mosquito-borne diseases, okay? And chikungunya might sound like it's a fancy name, but it's also very similar to dengue. It's just a little less severe. It is considered to be a hemorrhagic fever as well. So hemorrhagic fevers are the diseases that, like Ebola, they cause you to bleed out and die from that. It's less severe in the case of dengue and chikungunya, but they can cause hemorrhagic fever, okay? Chikungunya is endemic here in the United States. We actually expect several cases a year, and it is common in Texas, Florida, the deep South, okay? So these are diseases that never existed in the United States prior to the temperatures rising, and now they do. The mosquitoes that we have in the United States, whether it's Anopheles or Aedes aegypti or Anopheles mosquitoes, both of those can harbor very severe illnesses. Illnesses like malaria, which we may not have here in the United States, but just because it hasn't moved here. Another common one, and if you're from Ghana, then you might know this one. Another common one that is common and a big, big concern in cattle is Rift Valley fever. It is a massive issue in sub-Saharan Africa. These tribes, smaller villages, often rely on their livestock for livelihood. And Rift Valley fever is a viral disease that is 100% abortive in cattle. 100% abortive. Their cattle gets Rift Valley fever, they cannot carry calves. And it can kill the adult cattle too, but it's the fear disease. And I mean, I'm telling you, you guys have no idea this is going on, but everything that comes in through the United States through customs, that's why we check that stuff. And if you're bringing an animal in from outside of the country, they're going to quarantine it and check it. And the number one disease they're going to check for is Rift Valley fever. It's going to, well, rabies, we have rabies in the United States, so we don't need to keep it out, but we don't have Rift Valley fever in the United States, but all of our mosquitoes can harbor it. So if we get one animal in here in the United States that has Rift Valley, it gets bitten by a mosquito, and suddenly what's going to happen to the United States livestock industry? Yeah, can you imagine? And so that is a primary concern actually for biological warfare as well. But we consider agricultural warfare, agro warfare or whatever it's called. Anyways, so that we can keep in mind when we talk about rising temperatures, we're not just talking about the effects of how hot it is outside to us, the effects of the other organisms in the ecosystems as well, including diseases that are associated with warm temperatures. The diseases that we are commonly used to poo-pooing on these less developed countries, like in sub-Saharan Africa or in Central or South America, and thinking that we know better and we're just cleaner. But now that the warm temperatures are coming here, those diseases have nothing to do with having more money or anything like that. It just has to do with their climate. And so we're going to learn about that, unfortunately, the hard way. All right. They're all drawings. Now we're going to move into, take a break, take a stretch. We're going to move into chapter 7 next. I'm trying to get rid of this pen. There it goes. All right. Take a break, take a stretch, take a minute, and then we're going to do chapter 7, which is our viruses. So we're going to learn a little bit about viruses and whether or not we think they're alive. Go ahead and get signed into this Kahoot, and then we'll get going once everybody's signed in. Sit down. I did Pilates this morning on my back. Oh, I got that instructor that always wants to work out everybody's hips. And I had her 2 days in a row, you guys. My hips are killing me. Yes.
Like this coming Monday, now we have
It's a lecture exam. We only have 2 lab exams. They'll be midterm and final. So yeah.
Just to make sure there's no study guide for the lecture exam, but there is one.
Yeah, I mean, you could look at it that way. So I haven't named something a study guide, right? So yes, so the only thing that we have for the lectures is only, but we have the Bloom's challenge questions. Treat that like a study guide, like say that's a study guide, but they're a lot like the learning objectives, just like spinning a different spin on the learning objectives. So you can answer those as well as you can know the learning objectives. So that's the closest thing as I have to a study guide for the lecture.
Okay, so the 2 study guides on here, there's like 1 and 2, that's for the lab.
That's for the lab.
Okay.
Yes. No rush on necessarily studying for the lab one. Lab one isn't until October. Huh? It's like Bloom's challenge questions or something like that. We can look at that too. I know, because we kind of talked about it a little bit in lab, but let's, because I know we're getting close, we're actually thinking about it now. Let's go ahead and just talk about it really quick. Especially since, and I'm sure you guys are still thankful for it, you're always gonna be thankful for it, since today is gonna be a kind of short lab, okay? Don't tell my bosses. I had to have this discussion. I know I told you guys last time, like, I'm not supposed to be letting you guys go early, but I have changed. They changed my class. I say I've changed, but I am not the one that changed it. They wanted my labs to be longer, but they didn't really give me time to like remake the class. So.
Dang, we're not mad at you.
3, 4, 6. Right, yeah. I've actually already talked about it with my boss, which is Dr. Rhodes. I've told him, I was like, look, we're gonna get out of lab early this semester, like, yeah, right. Yeah. All right, so if you guys go to modules, it'll be in modules that you'll find the study stuff, okay? This is week 5. Is that 4 or 5? 4. Okay, thank you. Yeah, it's like, I don't think it'll really matter, but you'll notice that on the week, it says week 2, it has the date, so you can just find today's date if you needed it. So it is the 8th, so it's 8 through 12, and it says what's gonna be in there. So it's unit 1, chapter 5 and 7, and lab 5 and 6. And remember, you have your overview, which is your basic general stuff, and the lecture and lab section. Preview, which is our pre-lab or pre-lecture, again, if you're doing the pre-lecture for extra credit. Then what we do, the concept maps for both of them, that's our learning. And then our applying is our lecture application activity. Now, you guys don't probably usually see this because most of the time we do this in class and I'm going to put in the grade for it at the end of the class. So you don't usually see this. But it is here in the modules. If you ever wanted to click on these, this is where you'll find the sheet that I go over. It'll open up down here, but this is where we find that sheet that we're going over and the questions if you ever wanted to review them. And there's also a link to the Kahoot. Review the Kahoot quiz, this quiz that we're playing in class. So if you ever wanted to use that for studying, that's available for you in those. So this is in the modules. In the modules. All right. And then the quizzes, which we know, and then the review stuff. So I always have this block of the review stuff at the bottom of every week, okay? So there is audio shorts, which is literally if you click on this for unit 1, we have a short on germ theory. We have a short on selective and differential media, whatever. If you wanted to go all the way down to the eukaryote stuff, the different organelles, it's 10 minutes. None of them are really gonna be longer than 10 minutes. I try to keep them at 10 minutes or less. So there's one on viral structure and genomes, whatever. So if you wanted to hear, if you aren't good on certain things, just listen to a short about it.
Hang on. Is the exam equal each unit? Let me get that.
This one, I mean, I don't have it like laid out specifically equal, but I think the unit 1 exam is pretty equal between chapters. Pretty equal.
Earlier. The whole thing about going around like TikTok and social media about like everyone has worms. That's just
It's a lie.
so great.
Yeah. I can't with it, okay? I can't with it. And so listen, you'll have people that'll even be like, this is my poop. Like, and they'll show a picture of like, there's a worm shaped thing in there. And it's like, this is a worm. I'm like, that is your intestinal lining, sweetheart. And they're literally taking drugs or doing these cleanses and then they're pooping out stuff, but it's intestinal lining. Or if you eat a lot of fiber, fiber can often ball up and look like a worm in your poop. Now, if it is actively swimming around in your poop, then that is a worm. Congratulations. But if it is still a dead, that is not a worm. It's probably your intestinal lining. With how many people have to get colonoscopies regularly and things like that? Like, I'm pretty sure we would know if everybody had parasites all the time. All the time. Also, your blood work would show that stuff. If you have a high eosinophil count all the time, then you might have worms. That doesn't mean you have worms, but that's one reason why you might have. If you think you have worms, but your eosinophil count is normal, you do not have worms. You can't, unless you have some sort of severe illness with your immune system, which I hope you would know about otherwise. But yeah, if you're going around feeling otherwise fine, maybe a little bit tired, then you're a normal person on the planet existing in a first world country, okay? Let me tell you, when we talk about the helminth infection, so who in here, all right, let me think. I want to say this. What do you guys think is the most common parasitic worm infection? I'm just guessing. Most people will say tapeworms, right? Not even close. So the most common parasitic worm infection in the world is, well, it's made for animals, right? They might be heartworms, it's a pretty common one for animals get worms all the time, y'all. But in people, the most common parasitic worm infection is Ascaris lumbricoides, also known as the giant intestinal roundworm, which we don't even have in the United States. So that should put into perspective, if you think a tapeworm is common, or if you think pinworms are common, which are also common in little kids. Okay, little kids get pinworms, the itchy butthole thing, okay? Yes, if you have kids, you might know about this, or if you work in any medical setting around kids, and your doctor might even talk to you about this, but your kids, your butthole is itchy. This is, I'm not saying this to be rude, but it's a common thing. And kids playing, especially in sandboxes and things like that, okay? And do they wash their hands before touching their mouths or eyes or things like that? Probably not. So then what happens is then they go and whatever they're digging in, cats and other critters pooping in their like sandbox, treating it like litter box, get these worms in their bodies. Really, really actually pretty common for kids to have pinworms. And that is why ivermectin is sold over the counter, is you can buy ivermectin to treat your kid's pinworms. And again, it's kind of like lice. If your kid has pinworms, you have pinworms, and the whole family needs to be treated. Okay, so that's very common. Probably that's the most common one in the US, but it's still nothing compared to Ascaris lumbricoides, giant intestinal roundworm. Just real quick, because we're talking about the eukaryotes. Ascariasis is what we might call the disease caused by this. These are the worms. This is somebody's intestine that has the worms. These are, yes. So,
I have a fear of like a weird earworm. You really uncomfortable.
so these are real and these are not uncommon. And so the other thing is that these worms, the larvae migrate out of the intestine and through the circulatory system and then find their way into the lungs. And they will kind of mature in the lungs, and then you will cough them up and swallow them again. And then they complete their life cycle in the gut, and they can keep cycling like that. So oftentimes there are pictures of kids that are like coughing, worms crawling out of their mouths or their noses. I tell you, this is the most common of all the helminth infections in the whole world. So we're sitting here being like, what do you mean? I've never heard of this disease, because we're privileged Americans. But you go to a third world country. This is very, 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 very common. Okay, look at this. Yes, the distended stomach on the kid sometimes can be due to malnutrition, but it can also be due to worms in the gut. There's all sorts of reasons why you can have a distended stomach like this. But anyways, I want to bring attention to this. I just want to do this too, because most of us are going into healthcare. We need to get comfortable with seeing stuff like this. So when I start showing you pictures of genitals of people with STIs, that we're not going to be too shocked with that stuff, hopefully. Because man, when I tell you about chancroid, chancroid it gets me a little bit lightheaded. I'm not going to lie. All right. But anyways, study purposes for the exam. Back to this. Okay. Anyways.
Let's see the shake.
Oh,
Yeah.
okay. Because this is sexually transmitted. It might be safe images. It might be, yes, there's going to be quite a few.
Oh.
So anyways, these appear on the genitals. You might attribute some of these lesions look a lot like what we see with syphilis, but it's very similar. So the chancre that we see in syphilis is a hard nodule that's usually painless, but in chancroid, it's a wet, soft, very painful chancre.
Like herpes?
It's not, it's a bacterial infection caused by a bacteria called Haemophilus ducreyi. We'll learn about these. This is the chapter 24. This is literally our last chapter, okay? But keeping in perspective why we're learning about these things. Don't just always think about COVID and the flu and stuff like that. Like there's these other diseases out there that we might not see a lot. And we'll talk about like, you know, Ebola and stuff like that too. But I'm sure you guys have seen images of like heartworms and stuff like that. I'm not going to just sit here and show you gross images all day, but feel free to look it up, okay? And you will learn some of this stuff as we go through all this. But anyways, back to studying. So that's there. And terms, if you are a terms person, there's a list of the terms. I put those at the bottom. The Bloom's concept guide, think of it as a study guide, but again, it's very similar to the learning objectives. They're just different kinds of questions. Some of them might be very similar, but anyways, explain, hypothesize, compare and contrast, create a diagram, whatever. It's that sort of stuff. So this is as close as we're going to get to the study guide. And then there are relevancy links also. You don't have to look at these. These are just links where, like, if I really had a lot of extra time at the end of a lecture, I might play some of these videos.
Has been going on for billions of years that breeds well-armed monsters with other monsters for survival, having no particular interest in.
This is a video about the brain-eating amoeba. So if you wanted to watch relevancy links, it's just talking about diseases or something like that that's relevant to the chapter that we're doing. All right. Some of them are stuff like this, where this is Kyrgyzstan, which is a great resource for microbiology, physiology, like general biological sciences. They have really good videos and they're very well animated. Same like Amoeba Sisters, also a great resource. Okay. But anyways, there's a ton of them if you ever wanted to look more. Lissa virus, I don't even know. Okay, never mind. Nerding out. We need to stay focused. Anyways, that's all there at the bottom of the weeks. Okay, and so there's study resources there. Week 5 is when we have the actual exam. So what you will see for week 5 is that you really only have like study resources down here. Okay, so there's no relevancy links down here, but the audio shorts, the terms, and the Bloom's concept guide are all there for week 5. So those are there to help you, as well as, you know, in the discussions just while we're here,
Do you think if we
the weekly reviews.
like, do you think if I were to just like on the terms, if I were just to like write down definitions and then on that other one where it's the questions, the Blooms.
Yeah.
If I do both of those, I think that would just be great.
I wouldn't just answer them, right? Don't just answer them and then have the answers. That is not enough for this class. Like, you'll get a C, but you want to do better than a C. I'm telling you, that's how most people study. They're like, I answered these questions and I know what the answer was for that question. None of the questions that you're going to have on my test are going to be those questions, right? So you have to prepare yourself beyond that. So when you're doing that, I feel like the terms is a good thing to have just so you have the terms, right? Do flashcards for that. I'm not going to poo-poo on a flashcard for some terms, all right? But don't just do that, obviously, but then you're saying also answer the questions. If you're answering the questions, be able to explain it to me. So if I were to say, you know, go through the Blooms or the learning objectives and answer those questions, but then think of it as if I were to sit you down and the test was you, like I was to ask you the learning objective, explain it to me. Expect treating it like that, all the learning objectives. Treat it like you could explain it to me. That's the level of understanding I'm expecting. But those are resources that are probably enough to get you through, like just the terms. Get the terms under your belt. You got to have them under your belt. There will be some questions that'll be like, define this term. There will be. Some questions will be. And then other ones will be, you have to know it and be able to apply it. And I feel like if you can explain the topic to me from the learning objective, then you're pretty well.
So you'd say learning objective over the Blooms.
Yes. Well, I mean, the learning objective is what I write the test from.
Okay.
So yes. So I'd say learning objective. The Blooms is there as like a resource for you to test your understanding of the learning objectives. That makes sense. So yeah, see if you understand them well enough that you can explain the blah, 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 or make a concept diagram or whatever. Yes.
Quiz like that we're gonna be doing, like the big one, that should be that.
The Kahoot, I think it's gonna post. Let me see when it's gonna post. It hasn't posted yet. It's having an automatic post. You're gonna have to wait for me to see all these ones I have delayed until I have a lot of my posts are already programmed into the course. So let me see when it's gonna post. I feel like it's this week. Critical thinking questions post today at 10:30 here in 5 minutes. You'll have examples of the critical thinking questions. Week 4 review session. This will be my Thursday review thing. That'll be, oh no. Did it go down to the beginning? It did. I could search for it, but here it is. Week 4, this. Unit 1 review, Kahoot games. Okay, I will tell you that we will be having the review on Sunday at 4 p.m. Say 4 p.m. I said 8 p.m. previously, yeah? I don't care what time it is. You guys have a preference, 8 or 4 p.m.? We'll just do it at 4 p.m. because this is what the thing says, so I don't have to change it, okay? We'll be at 4 p.m. on Sunday, I guess. I don't know. I'm free on Sundays generally. So that's when we'll do that. Again, if you can't play it with us, there will be the link and it'll be click here to play it on your own time. Yeah, that will post on September 11th, Wednesday. Okay. We good? Okay, let's go on to chapter 7. Make sure you're signed in and we're gonna get going. We'll give you just 2 minutes to a couple minutes, make sure you're signed in. All right. What do we have? 1, 2, 3, 4, 5, 6, 8, 9, 12, 13, 14, 15, 16, 17, 18. Double check we're signed in. We're gonna get going. If you need to close out of the app and then re-sign back in, it's okay. All right, we're gonna get going, guys. Remember, you can always join in using the code. We need to go. All right, chapter 7, question 1. True or false? Antibiotics fight viruses. Is true or false? Okay. So this one is false. I feel a little dubious asking this question, all right? Let's see, is Kaya here now? No Kaya still? This person's not here. Sammy Adams. No? I feel like this is like out of order. Okay. Kylie Lippy. Okay. All right. We're gonna try. Antibiotics don't fight viruses. Okay, so I'm giving you that one. What do they fight? Do you think? I'll help you out. It starts with a B. There you go. Yeah. Okay. So they only treat bacterial infections. So I'm gonna help you guys out a little bit with this. Antivirals treat viruses. Antifungals treat fungi. Antiprotozoals treat protozoa. Antihelminthics treat helminths. But antibiotics treat bacteria. When we talk about something having antibacterial action, that's usually like hand sanitizers and things like that. But our drugs, antibiotics are what we call them. Don't ask me why, because science wants to make life harder for all of us, okay? I feel like sometimes with their words, but just remember that one. Antibiotics only treat bacterial infections. All right, true or false? Viruses enter host cells and use them to replicate, creating more viruses. Righty. This one was true. This is basically how viruses exist. The viruses, man, they are pretty wild. I don't know how else to describe them other than that. Where's my pen at? I'm gonna keep playing with stuff. Viruses really are just like two things. They only really have to be two things. A genome on the inside, okay, easy enough. Some sort of genome. I say some sort. We're gonna get to questions about this stuff. And then a protein coat. This is all that's required. Why are we emailing me right now? Okay, that's all you need. It's just a genome with some protein on the outside. And some of them also have a membrane on the outside, but they didn't make their own membrane. They steal it from the host. All right, so we're gonna keep that in mind as we're going forward. I'm gonna leave that up there. Not that it helps much, but it's something. Next question, what is the capsid around the viral genome made of? And Taylor, you've asked a few questions. I'm not going to call on you. I feel like you participated in them. You would have been the next one in case I didn't give it away. All right. So I said, yeah, so I just, in the drawing, we have the genome encased by the capsid. And that capsid is made up of protein. So it's a protein coat. Okay, so I'll even add that in now that we've had the question about it. Protein coat called the capsid. All right, who do we have? 1, 2. I only got 4, 1, 2. I'm not good at counting. Yeah, 5 more. Okay, so I'm not gonna ask a question on this one because I kind of went over it. Move on. We're getting our basics about viruses under our little belts. All right, this is a multi-select. Viral genomes. What can they be like? Choose all that apply to the possibilities of a viral genome. London Cooper, I'm gonna call on you. Okay. All right, so we pretty much got all this, right? The only wrong one was the brownish color one. London, why is that one wrong? There's a dead giveaway in the writing of this one.
Like in the question or like?
In this choice, why is this choice, oh my God, wrong? I'm gonna move this so we can actually see it. I'm trying.
There's no structure.
There's no structure. There's something else that gives it away. It says it encodes the virus's cellular structure and organelles. So organelles, we're getting there a little bit better, right? Even more general than that,
Viruses don't have, they're acellular.
they're acellular. They don't have cells. So already giveaway. This encodes, do the viral genome encode the virus's cellular structure? Ooh, guys, don't get tripped up on crap like that, right? It's in there right away. This is an easy, not this answer. At least if you can remember that viruses are acellular. They're not cells. Do you guys remember what cells are? All right, so I'm gonna call on somebody. Ooh. So I'm giving you points, London, by the way, because we did get the organ out. Okay. Lauren Singletary. Okay. All right. So what's a cell?
Oh, the basic building block.
Sure. So, so we know we're not made of living organisms, are not made up of viruses, right? We know we're made up of cells. So we can actually define it more specifically than that, and I'm gonna still give you points for it. But right, so it's the basic building block of life. A cell is a membrane-bound structure. The structure itself overall is membrane-bound, right? And it's capable of making copies of itself. It has everything it needs to make a copy of itself. Even your own skin cells can do that. You cut yourself accidentally when you're cooking, you cut your finger and it heals itself because your cells can self-replicate. But viruses can't do that. They have to use your cell's machinery to make copies of themselves because all they are is a protein coating around a genome. That's it. They don't have ribosomes. They don't have endoplasmic reticulum or Golgi or vacuole or lysosomes or mitochondria or chloroplasts. They can't do endospores. They're not being vegetative. They can do nothing.
How do they die when they're out of the body?
Mm-hmm. Yes. And so viruses are actually pretty weak. And many of them, especially the ones that have a membrane around them, they don't live very long outside of the body, typically. Okay. So the ones that have a membrane around them, they dry out pretty quickly. So usually they don't survive very well outside the body, but these other organisms live outside of the body, right? And multiply outside of the body, stuff like that. So anyways, good discussion on that. Let's go on to the next question. See what it has to tell us about viruses. Viral envelopes. What is a viral envelope? We talk about cell envelope, but for viruses, what's an envelope? They're not cells. All righty. It's the outer membrane structure that surrounds the capsid of some virus. This is our virus. Some viruses have a membrane around the outside. It's really bad drawing, but it's membrane on the outside. Think of it as this. The virus builds up its little genome. This is the viral genome, okay? Covers, I can't put the protein coat around it, so never mind, it's not a good example. It builds up its genome, and it builds a little protein coating around it. And then after it's done that inside of your cell, it pushes out of your cell membrane. We call it budding, okay? So imagine there's a big wall of blanket in front of me here, big blanket. And I just walk through it. I just slide myself in the blanket and walk through it. That's what the viruses do to your cell membrane. And so many viruses are enveloped like this. They steal the membrane from the host, just like that. They just push through. And they don't want all that junk that you have on the outside of your cells. I mean, you've got all these proteins on the outside of your cell that help with adhering to the cells nearby, adhering to your tissues, signaling, receiving things from outside channels and all that. They don't need all that on their blanket that they're coating themselves with. All they need on their blanket are the spike proteins. You guys remember about this when you're making your stuff, concept maps. All they need on the outside of their blanket is their spike protein. Their spike proteins are like keys that get into the next cell. And so what they do is, with their genome, they get into your cell and they force your cell to make their proteins, which is their capsids and their spike proteins. And those spike proteins just completely cover the outside of your cell. to the point of excluding any other molecule on the outside of your cell, right? And if I'm telling you there's molecules on the outside of your cell that do a job and they're meant there to do their job, right? And that's when we're replacing them with these viral proteins. Now I can start to understand why we feel like such crap when you have a viral infection. Because your cells can't stick to each other the way they're supposed to. And plus, there's a key on the outside of your cell now that unlocks the cell next to yours. And when that happens, when the key to the cell next to yours is studying your cells before the viruses butt out, you're unlocking the next cell and you start fusing with the next cell next to you. And then that cell starts making keys. And then it starts fusing with the cell next to it. And we get these big old, large, multinucleated cells. We call that a syncytium. When we have multiple cells, we call it syncytia. So it's when the keys, the spikes on the outside of one cell while we're making viruses, unlock the next cell and start fusing with it. So syncytia. Sound familiar? Anybody sound familiar? Anybody? Respiratory syncytial virus, RSV. Very common for viruses to do this. Okay, not all viruses do it to that high of a level. But also some viruses, they stud your surface of your membrane so much with those proteins, you can no longer adhere to the cells next to you. Imagine that this is happening in your blood vessels. That your blood vessels, the cells that create your blood vessels, can no longer stick next to each other. Now we have leaky blood vessels. This happens, of course, first in the capillaries, where it's only like maybe one cell layer thick. And so you start having these little bleeds under the skin. We call them petechiae. And we know petechiae are a common side effect of many viral infections. Even bacterial infections can happen, right? But petechiae, when we have a virus that does this to a large enough extent, not just causing petechiae, but even larger blood vessels eventually get affected. And now the blood can't be held inside the blood vasculature in general. You start bleeding out. Those are hemorrhagic fevers. That's Ebola, dengue, yellow fever, and chikungunya, and Lassa fever. Those are hemorrhagic fevers. Okay. Don't read too much into a hemorrhagic fever. Hemorrhagic means you're hemorrhaging, bleeding. Fever means you have a fever, elevated temperature. Hemorrhagic fever. So viruses have that much power just having a genome protected by some protein. Yes.
So the keys that you were talking about, would that play into the cytopathic effect?
Mm-hmm.
How it changed the pathology?
Exactly. So when we're having that key unlocking the cell next to it, and we have that syncytia, that's a cytopathic effect. The cytopathic effects, like she's talking about, basically are things that we can view at a microscopic level and see how the cells are being affected by the viruses. And that'll be things like cells fusing together or not adhering to surfaces like they're supposed to. Those are common cytopathic effects. Also inclusion bodies. If we're making just a buttload of protein to make new viruses, you'll have just a ton of protein hanging out in the cell, and we normally don't have that much protein structures. And these proteins are meant to basically stack up on each other. We've seen pictures of these guys. These guys are wild looking, right? They have like triangle shapes that they like stack up on each other. It looks almost like alien spacecraft and stuff. Well, those, we have to make them first in order to build the viruses, and they'll just accumulate in the cell stacked up like that, and they'll crystallize like that. And that's where we get inclusion bodies from. So all of those are cytopathic effects, things that we can see in human tissue when we look at it under a microscope. Okay. We cannot see the viruses themselves with a traditional light microscope. We can see viruses with electron microscopes. We don't just have those available and ready to use. You can't just make a tissue sample and then look in the microscope at it. It's a very, very complicated procedure and prep, and it's extremely expensive. So we don't use it as a diagnostic tool, but we do use it as a scientific tool. Okay. All right, moving on. I didn't ask a question about this one either, but we went and talked enough about it, so I think we're good. Let's move on. We got 3 people left. 1, 2, 3. All right, icosahedral capsids. Right. Icosahedral capsids is just talking about like icosahedral triangles. So it's even on all sides. That's what a icosahedron is a structure built out of icosahedral triangles. Okay. So icosahedral capsids, like isosceles triangles, is what I'm trying to say. Isosceles triangles, even on all sides. even on all parts. And 20 faces of triangles makes an icosahedron. Structure made out of that. Yes, they do look like that. And yes, they are actually shaped like that. It is wild, but that is how they do. And it's almost like a crystallization of protein surrounding their genome. Okay. There's not much I can ask about this. Not much I can ask about these things. Keep going. Often these are so weird to people. Bacteriophages are viruses. And they're involved in which of these processes? This is a difficult one for some people. It's in the picture, but like, I mean, yes. So this is transduction. Now, let me put this into perspective here, okay? Transduction is whenever a bacteriophage infects a bacterium. Bacteriophage is a fancy word for a virus that infects bacteria, just to be clear. That should be made clear whenever we were making our concept maps for chapter 7, okay? You'll often hear me talk about phages. A phage is just a short word for bacteriophage. And a phage is a virus that infects bacteria. Yes, viruses can infect any cell, even bacterial cells. So bacteria can be infected with viruses. And whenever this happens, the bacteriophage can incorporate its genes into the bacterial genes. That was a process to remember what we called this. This was lysogeny, okay, when that happens. And when that lysogeny happens, it can give new genes to that bacteria, and we call that lysogenic conversion. That process of having lysogenic conversion occur from a phage infection is called transduction. I know it's a lot of terms, but we got to know it. Okay. Transduction is the process of getting infected with a bacteriophage, having the gene give you new genes, and now you have a new gene expression. You might think like this sounds like, okay, so this is some random weird thing that can happen almost every single bacterial infection that you get. That bacteria got its toxins from being infected by a phage. Now this is getting meta, because I'm telling you now that the only reason that you have strep throat causes your throat to turn red and cause pain in your throat and it leads to potentially having a rash. How many of you guys got a rash whenever you had strep throat or had kids that got a rash that had strep throat? Okay. That is something that we would call scarlet fever. That is scarlet fever. That's what scarlet fever is, when you get a rash from strep throat. Okay. Scarlet fever happens and the throat pain happens because of the toxin that Streptococcus pyogenes makes. Streptococcus pyogenes makes that toxin because it got the genes for the toxin from a phage. That's lysogenic conversion. And that lysogenic conversion happened because of the transduction process. Yes.
See that transduction was what a bacteriophage, when you get the virus's DNA into your stuff?
When the bacteria is infected by a virus. The bacteria is infected by the virus. The virus incorporates its gene into the bacteria gene. And now that bacteria has new genes, like the toxin. Okay. Right. Most of the bacteria, like for example, Clostridium botulinum, botulism toxin, came from bacteriophage. It's crazy, right? And like so many bacteria that cause diseases in you, they were sick with a virus first. That's crazy. Did you know 30% of your genome is viral DNA? Huh? It might be higher than that. Yeah. This is like me having learned this in 2008 and I've retained this information. It's probably now higher than that. It's probably 30. It's probably closer to 45% of your genome is likely to have been from your ancestors got infected with viruses and now incorporated into your genome. That is wild, right? I mean, I feel like that's pretty impressive. Anyways, okay. So we've talked about some of these before, so I'm going to call somebody out relating to one of these, and I'm just going to ask you to pick one. Kay Sanders, are you here today? Okay, Kay. Good luck, it's okay. Pick, not transduction, we just talked about this. Any one of these, and can you tell me something about it? Even general stuff. Like what is conjugation? Or what is transformation? Or what is transcription? Transcription might be the easier one, but because I feel like you guys have talked about this in other classes.
Transformation. Wouldn't that be where the viruses maybe serve the virus?
Okay, yes, I'll take that. Because there's two different kinds of transformation that we will talk about in the class. I was thinking of the other one, but that is one that we have to know. There's two different ways that we'll use transformation in this class. One of them is if you get a viral infection and it causes your cells to multiply uncontrollably, then that's cancer. And so there's a term for that. Jackie, you're sitting right next to her. Can I call you Jackie? Is Jackie okay? Okay. Yes. So what do we call those viruses that can cause cancer? I'll help you. Starts with an O. I'll help you a little bit more. In hospitals, we have whole wards that are dedicated to this. So cancer, right? Onco viruses that cause cancer are called onco viruses. Just oncovirus. Yes. Okay, so we got there. We're getting there. We're getting there. Okay. So yes. And so don't be worried about that. That is literally what it sounds like. Oncoviruses are viruses that cause cancer, which I think is another question that's on here anyway. But yes, so you guys know this. Women should know this for sure about viruses that can cause cancer. Can you guys name one? Cervical cancer, excellent. So HPV, human papillomavirus, HPV. Now, when I told you guys that trichomonas was the most common non-viral STI, HPV, over 90% of all sexually active adults will have had HPV. Over 90% at some point.
Isn't it like forever?
No.
Oh.
So you can have it. So yeah.
Oh,
Yes.
gosh.
So HPV, human papillomavirus, some strains cause genital warts. Other strains might be associated with cervical cancer or other cancers. It doesn't just cause cervical cancer. It's also been linked to penile cancer, anal cancer, oral cancer, throat cancer, and certain like lower rectum cancers, rectal cancers. Okay, so basically sexual transmission of cancer in any sexual orifice. Anyway, okay. So HPV. This is why we do pap smears, right? Pap smears don't just detect abnormal growth of cells, they also can detect HPV and specifically the certain strains that are associated with cervical cancer. And so that's why that's going to be so important. So we're going to move on. I've called on, I think Isabella's the only person I haven't called on yet. So you know who's coming next, Isabella. Is Isabella here today? Yeah, you know you're next. Oh no, lysogeny refers to, I'll get you guys to answer this and we'll see if I can find a decent question. This stuff is difficult. I know it's a lot of terms. Lysogeny. All right, so this is whenever the viral genome inserts into the bacterial host genome. So viruses that are, so let me be more specific, phages that infect bacteria, right? Bacteriophages can basically alternate between, some of them can, alternate between the lytic phase and the lysogenic phase. Whatever. Those phages that can switch between lytic and lysogenic, we call them temperate phages. Okay, so temperate phages can switch between lytic and lysogenic. There are phages that are only lytic. There are phages that are only lytic. But if you're lysogenic, you're usually going to be able to switch between them. We call you temperate. Think of it as like a temperate climate. Temperate climates can switch between it being cold and warm. Okay, not just warm all the time or cold all the time. Temperate switching between. Okay. So think of them that way. They can switch between lytic and lysogenic. So this is going to be my question for you, Isabella. What is lytic cycle? So lysogenic is when we put our genome into the bacterial host. What is the lytic cycle? Destroys that. And so this happens anytime that you are a bacteriophage, they'll multiply and they burst out of the cell. That's how they'll live their life, okay? So that's their life cycle. They don't ever bud out like our viruses do. And eukaryotic viruses, they can bud out, they can push out. So we're going to want our last question. We won't do our activity today. So we spent all our time with our discussion. That's fine. That's the point of all of this. Okay. Oh, there's this. We have two more. That's okay. Lysogenic conversion. I think we just felt like we just did this. Oh, this is just the next step. Never mind. Lysogenic conversion. We'll answer this and we'll just go on. Good job. Do we have Sammy Adams, Kaya Hughes, or Saber Thompson here that I didn't get? Okay. So lysogenic conversion is when the lysogenic phase, you're converting by adding a new gene in. This is our last question for the viruses. Oncoviruses cause blank. You guys know this one. And then we will head up to the lab. If you want to review that worksheet, that second chapter 7 worksheet, you are welcome to do that on your own time. Or you can probably other classes, I might get to it. So I'll try to post a link to my recording from other classes. If we get to it. All right, great job. We'll see our podium and I'll see you guys up in the lab.
Don't be shy.
Yeah, what's up?
So did you see that the virus structure, it's like this, the spike is on the very outside?
Yeah.
It is just.
The spikes are more like here, they're like quite literally spikes. So. This would be your capsid. Your protein coding, right? Here's your genome, which could be RNA or DNA.
See what we're doing.
Positive, it's single stranded, it could be positive or negative, right? Okay, so there's our gist of that. And then on the outside, there will be an envelope, which is from the membrane from the host. When we bud out, and again, many viruses have this, not all of them. Flu, COVID, they all have membranes. They all have the envelope, okay? And then on the outside of that is the spike. It is quite like a spike.
And they're everywhere, not all one.
Yeah, but all over the whole thing, yeah. So around that whole thing like that.
Oh, because I got the animal.
Yeah, I mean, that's the only aspect I want to be sure that we were clear on, is that it's kind of stuck in the envelope. And if you don't have an envelope, so this is for our envelope viruses. We also have naked viruses, which are just the shell, the capsid with the genome. And then they'll have a spike also, but it's part of the capsid. That makes sense? They always have a spike, and the spike is the key
To adhering to the.
to adhere to the whatever the target cell is. Yeah.
So I don't know if that's the thing that you had to cover your job.
Yeah. Mhm.
Okay.
Yeah.
Okay.
Yeah, I'll be right up there.